Today, I'm excited to present to you the potential of marine macroalgae in combating the marine pathogens which have been isolated from infected adult Hippocampus kuda, or commonly known as the yellow seahorse. But before we delve into the significance of marine macroalgae, let's understand why seahorses are important. The seahorses are remarkable creatures that inhabit shallow coastal waters around the world. They are not only fascinating organisms, but also play a crucial role in marine ecosystems. Seahorses are considered as a flagship species of marine conservation issues. Their presence helps maintain the ecological balance of the marine environment. To ensure the conservation of seahorse species like Hippocampus kuda, attempts were made in mass rearing this species. Rearing seahorses in closed aquarium alleviates the pressure being applied to them. By doing so, we can protect them from habitat destruction, because there is a loss of seagrass beds and coral reefs which are vital seahorse habitats, and also prevent illegal trade due to the high demand of seahorses for traditional Chinese medicine. But there is a problem that arises when you try to rear seahorses in closed aquariums. An impediment to successful rearing of seahorses are microorganisms that serve as causative agent of diseases, leading to infections from marine pathogens such as Vibrio and Pseudomonas. Isolating the bacteria responsible for these infections is crucial for the following reason. We can identify the specific microorganism causing the infections. This knowledge is essential for understanding the pathogenesis of the disease and developing targeted treatment strategies. Morphological characterization, DNA extraction and barcoding via 16S rDNA bidirectional gene sequencing were performed to determine the causative agent, and 3 Halomonas species were identified. These species are gram-negative bacteria. Knowing the bacterial species allows us to implement preventive measures to reduce the risk of infections among seahorse population. Effective control strategies can be developed based on the identified bacteria's characteristics and mode of transmission. It is vital to explore marine macroalgae for their potential antibacterial activity against Halomonas species and other marine pathogens. Bacterial infections in seahorses, as well as in other marine organisms, have become increasingly challenging to treat due to antibiotic resistance. And this resistance arises from the misuse and overuse of antibiotics. Exploring alternative antimicrobial agents, such as marine macroalgae, could provide a sustainable solution. These seaweeds produce a wide array of bioactive compounds, including secondary metabolites, which may possess antimicrobial properties. These compounds have evolved as a defense mechanism for algae in the harsh marine environment and show promise in combating bacterial infections. Current research is focusing on evaluating the potential of marine macroalgae, Sargassum crassifolium and Ulva intestinalis, against Halomonas species isolated from infected seahorses. Phytochemical screening of the crude ethanolic extracts of the 2 marine macroalgae was performed. Also, these extracts were then used for the Kirby-Bauer disk diffusion assay against the isolated strains of bacteria from the epithelial surface of the infected seahorse. The resulting plates were incubated for 30 degrees Celsius for 24 to 72 hours. Photographs were taken for all the plates. The photos were analyzed using UTSCA image tool software to measure the zone of inhibition. The mean diameter in millimeters and standard deviation were generated and recorded by the software. Past software was used to analyze the data. One-way ANOVA was used to check the statistical difference between groups of seaweed extracts tested into different strains of bacterial species. Tukey's pairwise comparison was done to check which concentration of algal extracts was highly significant. Understanding how the bioactive compounds derived from marine macroalgae act against Halomonas species is crucial for optimizing their efficacy. By elucidating the mechanisms of actions, we can enhance their antibacterial potential and minimize any potential adverse effects. The different concentrations of the algal extracts of Ulva intestinalis and Sargassum crassifolium showed inhibition on the bacterial strains of Halomonas species, albeit not comparable to the commercial antibiotic tetracycline. Despite the small zones of inhibition, the findings were still important since these seaweeds show potential source of antibacterial components. The presence of moderate amounts of flavonoids from the two macroalgae are responsible for the zone of inhibition, given that the extracts yield zones of inhibition despite the percent concentration of the extracts is very low. The successful identification and utilization of marine macroalgae with antibacterial activity can have significant implications for seahorse conservation. By incorporating marine macroalgae-derived compounds into treatment protocols can expand the range of available therapies for seahorse infections. This approach may provide alternatives to traditional antibiotics, which could be less effective due to the bacterial resistance. By harnessing the natural properties of marine macroalgae, we can develop sustainable and environmentally friendly approaches for managing seahorse health. This would reduce the reliance on synthetic chemicals and contribute to the long-term conservation of seahorse population. It is perceived that the presence of phytochemical components such as alkaloids, glycosides, flavonoids, saponins, steroids, and tannins exhibit different biological activities and are important indicators of the microbial properties of seaweeds. Herewith, macroalgae can be used as an alternative source of antibiotics that could treat diseases that are caused by marine pathogens in seahorses, hence can be valuable in aquaculture industry.